everyone, for learning outcome number 2 of Module 10. We're going to be talking about the functions of the small intestine, more specifically about the different cell types and what they secrete, and therefore what functions they exert within the small intestine. We'll start with the enterocytes, which are about 80% of the total epithelial cells. They're mainly going to be located right over here on the upper part of the villi, as we can see over here from this image. And the main function of the enterocytes is to absorb the molecules from the gut lumen and transport these molecules or nutrients toward the surrounding connective tissue and blood vessels. Therefore, these are also known as absorptive cells. In addition to the absorptive functions, these cells, they also secrete nutrients and salts as well as water in order to maintain the normal salt and water balance within the body. Now remember that water follows sodium, therefore if those cells are secreting salts, they are also releasing water. And the nutrients that they absorb will then be secreted into the surrounding blood vessels and taken throughout the body to nurture all other cells in the body. Now, the next type of cells that are also located in the apical part of the villi are the goblet cells. And these are cells that secrete onto the internal surface of the intestine a coat of mucus that will lubricate the chyme, and it also forms this protective barrier that prevents the enzymatic digestion of the intestinal wall. Now think about this. The small intestine receives all these enzymes coming from the stomach, through the pyloric sphincter, and also from the pancreas through the sphincter of Oddi. And if its digestive wall was not protective with this coat of mucus, then the enzymes would be digesting the actual digestive wall. And in fact, there is more mucus secreting cells in the duodenum than in any of the other two portions of the small intestine. And this makes sense since the duodenum is where most of the enzymes are being released into. At the basis of the villi, we find these entrances to what we call intestinal glands or intestinal crypts, or also crypts of Lieberkühn. And these pockets, they extend deep into the underlying lamina propria, as we can see over here. And it's in these crypts that we will find these specialized cells that will release a lot of the hormones that will help with the digestion in the small intestine. So the intestinal glands will contain these enteroendocrine cells that are going to be responsible for the production of several of the intestinal hormones that are going to be secreted, and also enzymes that will have important antibacterial activity. Now, there are several types of enteroendocrine cells, and most of them are going to be located in the duodenum, as most of the functions occur in the duodenum. So let's talk about each of these cells. First, we have what we call the G cells. These we already saw that they are also present in the stomach, and they will secrete the hormone gastrin in response to the presence of chyme in the stomach. And remember that in the stomach, it stimulates the release of hydrochloric acid by the parietal cells. And also in the stomach, but now in addition in the duodenum, it will also stimulate muscle contraction. And it makes sense. Since this hormone is released in the presence of chyme, then it will help in the digestive process. Next, we have what we call the I cells. These will secrete cholecystokinin, or also known as CCK, and this hormone will act on the pancreas and on the gallbladder. On the pancreas, it will stimulate the secretion of the pancreatic juices, which includes the enzymes that will help with the digestion of carbohydrates, fats, and proteins, in addition to the release of bicarbonate ions that will help to neutralize the acidic from the chyme that is coming from the stomach. In addition, it will stimulate the gallbladder to release bile in the presence of fat that's also coming from the stomach. And we will see how this will help with the digestion of fat once we talk about the liver. In addition, CCK also regulates the gastric emptying. And how does it work? Well, when the chyme coming from the stomach is not well broken down, or when the duodenum is still busy processing the contents that already came from the stomach, then CCK basically tells the pyloric sphincter to stay closed until the duodenum is ready to receive more chyme. And this also gives the stomach more time to process the gastric content and break it down even further to help the small intestine with the digestion and absorption process. In addition, CCK suppresses your appetite while you're digesting by making your stomach feel physically full and by activating the vagal nerves in your stomach wall. What does this mean? Well, the term vagal nerve activation comes from the vagus nerve, which is one of the cranial nerves. In fact, it is cranial nerve number 10, which carries signals between your brain, your heart, and your digestive system, and it will play a key role in your parasympathetic nervous system. And since the parasympathetic nervous system controls the rest and digest functions, when stimulated by CCK, it suppresses your appetite. Lastly, CCK also triggers bowel motility, which, as we know, contributes to muscle contraction that moves food along through your intestines to continue this digestive process. Next, we have what we call the K cells, which secrete the gastric inhibitory peptide in response to chyme that's entering the small intestine. Now, it receives this name because initially it was thought to decrease the gastric acid secretion. However, recent studies do show that its main effect is by stimulating insulin secretion in response to a meal. Next cell, we have the M cells, which secrete the hormone motilin, which, like the name suggests, will accelerate the gastric emptying, stimulate intestinal peristalsis, and therefore this will help the food to move from your small intestine into your large intestine. And it also plays an important role in controlling insulin release and triggering hunger cues. If you have low motilin, you may have slow or delayed muscle contractions in your small intestine. Lastly, it stimulates the production of pepsin, which is an important enzyme for protein digestion. Next cell are the S cells, which secretes the hormone secretin. Secretin is secreted when stimulated mainly by acidic chyme that's coming from the stomach. So if you have a pH around 4 already, then secretin gets released. Now, it will have this osmoregulatory function where it will regulate water homeostasis throughout the body. It also influences the environment of the duodenum by regulating the secretions in the stomach, in the pancreas, and in the liver. Therefore, it has this inhibitory effect on the gastric emptying and motility. Now, because water is able to pass through a closed pyloric sphincter, this is how secretin will be able to have this osmoregulatory function by allowing the water to pass through a closed pyloric sphincter, even though it will control the emptying of the gastric content. Also, in those crypts, we have what we call stem cells or undifferentiated epithelial cells that will line the intestinal crypts, and they will renew the mucosal epithelium by dividing very rapidly and moving continuously onto the villi. Now, these are going to be among the most quickly dividing cells of the body, completely renewing the inner epithelium of the small intestine every 3 to 6 days. This rapid replacement is going to be necessary because there are going to be so many enzymes that are present in the small intestine, together with the low acidity from the chyme that is going to be coming from the stomach, that these cells need to be rapidly replaced to be able to function properly. And the last type of cells that we find in these crypts are the Paneth cells. And these are epithelial cells that secrete certain enzymes that will actually destroy certain bacteria, and they may help to determine which kinds of bacteria will live in the intestinal lumen. So let's cover them a little bit more details in the next slide. The permanent bacterial residents of the intestinal lumen, they're called the intestinal flora. They are important for two main reasons. One, they control the population of what we call the undesirable bacteria by basically creating this survival competition between the healthy gut flora and the invading bacteria, and most of the time the healthy gut flora wins. And the next function is that it's going to be responsible for the manufacturing of some essential vitamins which the intestines are going to be absorbing. One of the examples can be vitamin K, which in fact it has been estimated that up to half of the daily vitamin K requirement is actually provided by the gut bacteria. Vitamin K has several important functions within the body, and some of which are still being discovered, but for now, researchers have shown that vitamin K is an anti-calcification, anti-cancer, bone forming, and an insulin sensitizing molecule. In addition, these gut bacteria produces thiamine, folate, biotin, riboflavin, and pantothenic acid, which are all water-soluble vitamins. Now, thiamine, riboflavin, pantothenic acid, and biotin all belong to the group of B vitamins, also known as B complex vitamins. In fact, thiamine can also be called vitamin B1. Riboflavin is also called vitamin B2. Pantothenic acid is vitamin B5. And all B vitamins help the body to convert food, mainly carbohydrate, into fuel or glucose, which will be used to produce energy. In addition, it will help to metabolize fats and proteins. Therefore, the B complex vitamins are necessary for a healthy liver, healthy skin, hair, and eyes, and they also help the nervous system function properly. Next, we have folate, which helps tissue grow and cells work properly, and together with vitamin B12, which will help to break down the folate, both will work to create new proteins and form healthy red blood cells. And we all know that for vitamin B12 to be absorbed by the body, we need intrinsic factor, which is released by which cells? Well, if you said parietal cells of the stomach, you are correct. Now, the lamina propria of each villi contains an extensive network of capillaries that will absorb and carry these nutrients to the hepatic portal circulation. And we will talk more about the hepatic portal circulation when we cover the liver. Now, in addition to the capillaries and the nerve endings, also notice the presence of the lacteals in the villi. We have already talked about the lacteals when we discussed the lymphatic system, as these vessels are part of this system. They will be responsible to transport materials that cannot enter local capillaries, and these materials, such as large lipid protein complexes, they will be sent through the thoracic duct and end up between the left internal jugular vein and the left subclavian vein, where it enters the blood circulation, whereas the end products of the digestion of carbohydrates and proteins enter the blood capillaries that are also present in the villi, as we see on this image represented here by the red and blue structures.